So hello and welcome. I'm Art Blank. I'm President and CEO of MDI Hospital. And this is the second in a series of virtual town halls that we're hosting. And our focus is to keep our community engaged and informed about COVID-19. As of today, the Maine CDC is reporting 4,368 cases of COVID-19 in our state, which also reflects 46 cases that have been diagnosed so far in Hancock County. Johns Hopkins is reporting 5.7 million cases in the United States and 23.7 million cases worldwide. So in our lifetime, none of us have seen a global crisis of this scale. And as we all are working to navigate this together, we are very fortunate to have those among us who have stepped up to lead the way. And today, I am very privileged to be joined by two of such individuals, Dr. Jonathan A. Epstein of Penn Medicine and our own Dr. J.R. Krevans, Jr. We have the benefit today of Dr. Epstein's expertise thanks to our 10-year partnership with Penn Medicine. In addition to connecting our community with leading experts, this partnership has allowed us to bring medical residents from Penn to Bar Harbor for 2-week rotations at MDI Hospital. And to date, nearly 100 Penn residents have experienced our island hospital environment firsthand, delivering care alongside their MDI hospital colleagues. As Dr. Krevans can attest, this partnership has introduced these urban trained residents to a completely different way of life and a completely different way to practice medicine. Over the years, our collaborative efforts have also brought several new life-saving procedures, equipment, and technology to MDI Hospital and enhanced our practice of evidence-based of both emergency and psychiatric medical care. And in fact, last year, a Penn Med resident decided to launch her career at our behavioral health center. And Dr. Rebecca Villarreal joined our medical staff as a full-time psychiatrist. Given the partnership, has increasingly had impact upon our care, and the waiting list of Penn residents who want to take part, we anticipate that more program alumni will join our staff at the hospital in the years ahead. And in these unprecedented times, we are more grateful than ever to have a decade of partnership with a world-class institution to draw upon. And we're also very thankful to Dr. Epstein for joining us today to share the work that he and his colleagues at Penn are doing to combat COVID-19. As we work to address the COVID-19 pandemic at the local level, MDI Hospital has focused on providing as much information, education, and support to the public as possible. In partnership with the Down East COVID-19 Task Force, we've established a pilot program for asymptomatic workforce testing with the hope it can serve as a model for other seasonal communities. And we've also launched a medical grade masking education and distribution project to ensure that all area businesses have the tools that they need to provide the highest level of protection possible. In early August, when calls from visitors who had received positive test results only after they arrived in Maine began to increase, we published a public service announcement to alert our community to the potential of unreported cases. This led to a very positive discussion with our state officials around a new contact tracing partnership that will ultimately grant MDI Hospital access and public health jurisdiction to track and trace cases such as those using the SARA alert system. To date, we are still working to finalize the details of this contact tracing partnership. And so because of that, we're not today going to spend a lot of time on that particular topic. but we're very, very appreciative of the work that our state officials are doing with us to get that project online. But we will discuss everything from vaccines to testing and new therapies to rapid testing, outbreaks, and your questions. Some submitted in advance and some that we hope and expect you'll ask today. So if you would like to ask a question during the discussion, please use the Q&A feature located at the bottom panel of your Zoom screen. We will answer as many questions as possible, time permitting, and those that we do not have time to address, we will follow up with you directly using email. So thank you all for joining us today. And now we're going to take a few minutes to introduce ourselves before we get started. So as I have introduced myself, I'm the president and CEO of MDI Hospital, and I just wanted to let you know that our hospital, for those of you, many of those of you that are in our community, how much we appreciate the work that you've done to help keep our community safe. There's, as we go through our daily lives, it is very reassuring to know that the commitment that our communities have made to safety. So now let me ask Dr. Epstein to introduce himself.
Art, thank you very much, and thanks for having me today. It's really a pleasure to join this group, if virtually. I'm a cardiologist and a lab-based scientist. I had my first exposure to lab research here on the island at the Mount Desert Island Biological Laboratories, and I'm very grateful for that exposure that I had. I did my training at Harvard Medical School and at Brigham and Women's Hospital before moving to the University of Pennsylvania some 25 years ago, where I've served as the chair of the cell biology department, a cardiologist, as I mentioned, and most recently as the chief scientific officer and executive vice dean at the School of Medicine. And in that role, I oversee a large research portfolio. Penn has amongst the largest of academic medical center portfolios, amounting to about $1 billion a year in a diverse area of research. And in particular, we pride ourselves that we focus on translational research and bringing new discoveries to have real impact. So we've had 11 FDA approvals of new drugs in the last 2 years or so. And that means we feel a particular responsibility to play a role in a pandemic such as this to really try to make a difference. And I look forward to trying to summarize a few of those areas of investigation on today's call.
Thank you. Dr. Krevans?
Hi, I'm J.R. Krevans, Jr. Some of you know me. Also started here on this island with a lot of my education, including actually finished my undergrad physics thesis on this island. Took a very different path. Fortunately, I'm actually the last few months been very glad for having done my undergrad in physics, because so much of what I've been called upon to look at in the last 5 months related to this outbreak, 6 months now, has been related to that. Went to medical school at UC Davis and then did my residency at UC San Francisco, and then spent 21 years with the United States Public Health Service as a medical officer, primarily doing clinical care, but quite a bit of laboratory medicine and epidemiology, involved quite a bit with in PHS with outbreaks of tuberculosis and RSV. And actually during my training, I was there basically at the start of the AIDS outbreak in San Francisco. So I'm not one of the people who is deeply shocked at what has happened this year. It was just a matter of when and subtle details, not if. And I've been here on the island for 15 years now, and back full-time, and I'm a hospitalist at the MDI Hospital. Previously was the medical director of ER, but I'm also the chairman of the Infection Control Committee, and I'm on the Safety and Emergency Preparedness Committees. And basically since January, this is most of what I've been doing.
Thank you. So again, thank you both for joining us today. Let me start by asking kind of a broad question, but to get things going, Jonathan, could you tell us about the efforts at Penn Medicine in research to combat this disease?
Sure. So let me just give you a sort of broad summary of some of the areas of effort that have been underway. You know, back in March, which seems like such a long time ago now, we learned of the emerging pandemic, as everybody did, and we immediately stood up a coronavirus research center to help to coordinate our effort. And one of the lovely things about having such a diverse portfolio and such a wide number of investigators is that it turns out we had a series of people who've been studying coronavirus their whole life. Susan Weiss, for one, has been an investigator in the field for 40 years and stood up to be one of the co-directors of this new center. You may have seen her on the platform with Tony Fauci and others talking on this topic. But the center immediately focused its research in four areas. And those broadly were testing, something we've heard a lot about and we still need to make more progress in. Happy to talk more about. The immune response to the disease, which includes antibody or serology testing, but also how different people seem to respond differently to the infection. Some don't even get symptomatic. Their immune system is doing the right thing. And we need to study those people so we understand what's right about their immune system. And some get horribly sick and end up in the intensive care unit with associated overactivation of their immune system. And we still need to learn more about why their immune system seems to go awry, but we've made some progress there. The third area is therapeutics. We realized immediately that we weren't going to have a vaccine right away, and the best bridge until we can have a good vaccine in place would be to have some medicines that could treat the disease so it wasn't so terrible or so lethal in some cases. And I often think about the parallels to HIV/AIDS in that regard. It's a little scary to think we've been working on a vaccine for AIDS for some time without success, but we have developed a armamentarium of antiviral agents, in particular a cocktail of agents that when used together really transform the disease from what it was when I trained, something really terrible, to something that people can live their life with now. And so we need to develop a similar array of medicines, and my own feeling is that it's going to take more than one medicine, but a combination of medicines that are likely to be able to treat this disease. And again, I'm happy to talk about some examples about how we're trying to find those and test them in clinical trials. And then the fourth area, of course, vaccines. And in this, Penn scientists have been deeply involved. 4 of the major lead vaccine candidates were developed in part or wholly by investigators at Penn, including the Moderna vaccine that you may have heard a lot about. It's based on stabilized messenger RNAs that Drew Weissman at Penn really helped to develop, and a DNA-based vaccine developed at the Wistar Institute that's affiliated with Penn, that's also being brought forward by Inovio, and a adenovirus or modified adeno-associated virus vector that Harvard and some private companies are bringing forward as well. Some of the phase 1 and phase 2 clinical trials have been underway and completed, and we've been very hopeful about some of these lead candidates. Again, happy to circle back on those. I will just comment, we also have a very robust clinical research infrastructure for doing clinical trials. And it's been remarkable to see how many people have pivoted their entire research efforts to address this problem. My own lab has changed completely to a COVID lab, even though we're fundamentally a cardiac research laboratory. And we can see that again and again and again across the institution. And one of the initial efforts that I think made a difference was quickly contributing to the remdesivir trials, which is one of the 2 agents, remdesivir and dexamethasone, that have really shown benefit in at least some patients. To my mind, only marginal benefit, but it's a beginning. And as I say, I think in the end, it'll take a combination of these medicines to make the biggest difference.
So I know as their efforts are varied in terms of the different kinds of vaccines that folks are looking at, and I know that Penn researchers are also looking at gene therapy approaches. Can you talk a little bit about what advantages they might have over conventional vaccines?
Yeah, so this is one of the areas that I'm most hopeful about, and it's really a somewhat unique approach. So it so happens that two of our leading areas at Penn are cell therapy, that you may have heard a lot about, immunotherapy for cancer, and gene therapy, where we can create treatments that replace a missing gene in people who have inherited disorders. For instance, if you're missing a gene important for vision, kids can be born blind, and we've had success in replacing that missing gene in the eyes of young children and restoring sight, really remarkable cures. So like everybody else, these groups have pivoted to address the COVID pandemic. And Jim Wilson's gene therapy group has proposed that to use the same viral vectors that deliver the missing gene to instead deliver the coding sequence for an antibody that would block the virus. So this is sort of halfway between a therapy and a vaccine. The idea would be to insert the DNA coding sequence for the antibody in a viral vector, inhale the viral vector through your nose into the cells lining the respiratory tract in the lung, and have those cells become factories for producing the antibody that would then coat the airway and prevent the virus from infecting the person. And they're teaming with Regeneron, who has sequenced some of the best antibodies from patients who've been infected. So we know the sequence of those antibodies and can insert them in the viral vectors. And this approach should be starting clinical trials at the beginning of September. One advantage is that if it works, we should be able to get the answer relatively quickly, within a month or two of clinical trials, get a hint of whether it's working. And the manufacture of such a product could happen quicker than some of the vaccines that may take a little longer than that to manufacture at scale. So this is something he's called BioShield, and I'm pushing it along as fast as we can. We're quite excited about it.
Sounds very exciting. It's very promising. Switching gears for a minute, there's been a great deal of conversation around the need and the importance of testing and contact tracing as a means of identifying potential infection and mitigating the spread. JR, could you talk about the number of rapid tests that we've heard about and how that might contribute to this goal? You're on mute, JR.
I guess I'm going to start by talking about one kind of rapid test, which
Yeah.
is frequently marketed, which is of no particular benefit. And we've had issues with this being marketed here in Maine to visitors, and there was a big issue at Sturgis, the motorcycle rally, where people are marketing the finger stick antibody tests as a rapid test for COVID. And there's basically two problems with that. One of which is many of those tests are terrible in their own right. They are just plain wrong. They say yes when they should say no, or no when they say yes, should say yes. But the bigger problem actually is they're not answering the right question. An antibody test says, does your body if this test is accurate, does your body recognize COVID-19? Meaning you've been infected with it at some point in the past and have responded to it. So early in infection, when you are most infectious, your antibody test will be negative. I mean, it's as simple as that. You get a test, it says no, but the actual answer is yes, you're infected. So antibody tests, which are the either a finger stick or a regular blood draw test, have uses primarily for global study of population. How many people in New York have been infected? If 1,000 people are infected, how many of them will die? Those kinds of things. Not much use for individuals. The other kind of rapid test, which has a lot of promise and is being utilized, are tests that recognize antigens, as opposed to a PCR test, which recognizes the actual genetic material, the nucleic acid, which takes more work to do. It's literally a cooking process, and it is done on a complicated instrument, and it's done with a series of heating and other chemical processing steps. Takes time. You can recognize proteins with simple, either visually read tests or tests that are rapidly read by a color reader on a machine. The problem is they are a little bit less sensitive, meaning there are cases where if you have a small amount of the virus in your throat or nose, the PCR might spot it while an antigen test might miss it. And they are probably less specific, and there's a lot of fight about that, meaning if the antigen test says you have it, do you really have it? So as far as the sensitivity, there are advantages to a test. If somebody comes into the hospital, to know right now or within 20 or 30 minutes, do they have COVID, is really useful. And interestingly, if you have a good test that is well run, while it is perhaps a little less sensitive, the people it misses are generally people who have small amount of virus, which doesn't necessarily mean they're less sick, but it means they're less contagious, which is often what we want to know. And you can follow it up with a PCR. The specificity question is interesting. There's been a whole series of interesting news stories ranging from a town in Vermont which thought they had an outbreak of 50 people, to a camp in Maine that closed, to a governor of Ohio who made national headlines for a weekend, where the antigen tests were positive, but on retesting with PCR testing and also simply looking at, gee, did any of these people spread it to anybody else? Did not show to be as positive. And I think that's acceptable as long as you know what you're getting. People sort of, when they get a yes/no answer from a test, think it's an absolute, while these tests are predictive, but they're not absolute. None of these tests are absolute. But there's the possibility that some antigen tests could be done very cheap and very fast. And I know there's a bunch of work going in various places, which could, for example, enable us to test everybody with any symptom, or even every school teacher every day, perhaps. Jonathan, you're working on some stuff like that at Penn, correct?
Yeah, we certainly are. We have efforts in a number of different areas for test development, and I just want to underscore much of what you just said, JR. The antibody tests, the serology tests, are not useful for determining if you have the disease or not. And they're mostly useful for testing a population to see how many people have been exposed in the past, but not so good for making individual diagnoses. And there's not much reason for an individual person to go to their doctor and say, I want an antibody test. There are particular cases where it might be useful, but broadly speaking, not so much. But the acute viral testing is very, very important, and we need more of it in this country. We've been testing thousands of people a day in the Philadelphia region, but we're handcuffed somewhat by supply chain issues where we simply can't get enough of all the various reagents necessary to do the testing. And so we need to develop more and diverse ways of testing. And in that sense, the antigen tests, the tests that measure the protein on the outside of the virus itself, can be useful because they're additional tools that don't rely on the same supplies as the PCR testing. Even if they are in general a little bit less sensitive, they're still likely to catch most of the people, especially, as JR says, most of the people who are contagious. So there's definitely a use for these antigen tests. At Penn, there are a number of cool things in development, and a number of companies we're working with who are developing home tests, like a pregnancy test, where you might dip a dipstick in your saliva and see if you're positive or negative. That's where I think this is going. And I think within the next few months, we'll start to see many more of these coming out on the market. The trick will be to be sure that they're validated well, because many of the tests that have been coming out, especially for the antibodies, the serology tests, were not well validated, sort of the Wild West. And some of them just were no good, and people were getting misleading information. So we hope that there'll be better oversight on some of these point-of-care diagnostics and home tests. We're working with two companies, OraQuick in the Philadelphia region that makes pregnancy tests and have shifted their efforts to make a COVID test, and Homodeus, begun by a brilliant engineer, Jonathan Rothberg, who developed next-gen sequencing that allowed for the sequencing of the human genome. He's put his efforts towards making a home test. And these, to me, both look quite promising, but still a month or two off, I think. We're internally at Penn trying to develop a test on saliva that is a little different than RT-PCR. It works at a different amplification method to detect the RNA of the virus. The COVID-19 or SARS-CoV-2 is a RNA virus, meaning it's a protein capsule packed with RNA on the inside. And this test uses something called RT-LAMP, a different amplification method, which is useful simply because it doesn't use the same reagents as RT-PCR. And we're literally making our own enzymes in the laboratory, so we don't have to buy them from anybody and won't have trouble having enough supply. And we're hoping to test all of our employees and trainees as often as we possibly can, every other day or twice a week, if possible. And I do feel like we need to get to the point where surveillance testing of this sort, even in asymptomatic people who are at risk for spreading or for coming in contact with others who might spread, is going to be important until we can get a vaccine up and going. And the best modeling data I've seen has suggested that even if the tests lack a little sensitivity, if you can use them more often, you'll catch people before they become and when they're infectious. And that's because the amount of virus in people's saliva or their other secretions skyrockets during the peak phase of infectivity and then falls off quickly. And so if you happen to test them before or after that peak, you need a test that's really sensitive. But if you're able to test them frequently, you'll catch it when it's peaking, and that's when it's most important to catch it. So we're trying to focus on frequent testing at low cost that would be relatively sensitive. There are many other groups working on this, as you all know. Yale just published something called Saliva Direct, which is a protocol for testing using PCR on saliva. And I want to call them out because they didn't team with a company and try to make a lot of money from it. They just did it for the good of the country, and they put the protocol out for anybody to use. And their protocol does allow testing at very low cost. Just to give you an example, insurance will usually pay up to about $100 for a PCR test. And the real cost of different protocols might be from $60 to $120 for the test. If you want to test everybody twice a week in any large population for 6 months or a year, you're talking hundreds of millions of dollars very quickly. So if we can drive that cost down below $1 a test, that's going to be just absolutely critical to make this sort of surveillance testing feasible.
So thank you, because you remind me greatly of a lot of wonderful conversations that we've had in our COVID task force, and with JR and Dr. Julian Kufler's guidance, emphasizing the need for as much surveillance testing as possible, and that has helped us to establish the asymptomatic testing pilot that's operating in our community. JR, I did want to make sure we got a chance for just a minute to talk about one of the other things that we've talked about in the task force, and that's some of the public conversation that's recently taken place around the different types of face coverings that are available, and some concerns that folks have expressed. Could you clarify that for us? You're on mute again.
All right, I could try. I guess one of the things that whole thing points out is, I mean, this is evolving science, and I think the reality is that most of most people in this country are not used to the way seeing science happen in real time. And those of us who are used to that are not used to seeing science happening at 60 miles an hour, which is what we're doing. The good thing is, though, that a lot of the stuff is available very fast. So what actually happened with that, and I think it's worth reviewing that study, is somebody came out with a study. It was actually a fairly decent study for what it was, where they basically looked at a little over a dozen different specific masks. They just took a bunch of things they could buy, and they put it on a mannequin, and they did some fairly good physics measurements of the particles that came through the mask in terms of size of the particle, number of the particle, distribution of the particle. And they said this mask, which we'll say brand A, worked this well, and brand B worked this well. And then there was a specific finding around a single specific commercial product, a single buff type mask that was a pull-up mask, neck gaiter type, that was a very low performing event. This then got reported as neck gaiter or buff type masks don't work, which was an over-reporting of what the article said, and the author tried to sort of talk people, reporters back through that. And then there's now been subsequent work sort of correlating what he did using a wider variety of products. And the reality is, what we know is that anything certainly with 2 layers of fabric that are at least moderately tightly woven works. The better it fits to your face, the better it works. It's like this is actually not surprising stuff. And it has to be comfortable to wear, or people won't wear it. Unless you're in an industrial or hospital setting where you will wear your 3M face clam or else. And so it's sort of, I think, tough public information to say. It's like there is no national standard. We are all being asked in our daily lives to do things that are normally an industrial hygiene thing, where your boss provides a product and says, this has been tested and validated, and here's your 30-minute training on how to use it, and then checks up on you. But we are, every single one of us, in order to go to the store, are expected to do that, and that's tough. The current data says actually that if it's a 2-layer buff, it's actually pretty good, because it turns out those fit really well. The fabric may not be quite as good, but the fit is better. It's not a simple question. But each of us can make the choice to look at what we're putting on our face and how effective it is. And there really is pretty good data that if it's a single layer thin buff that you can sort of see your fingers through, it probably doesn't do much. And I also want to extend that to the whole question of face shields, which was heavily promoted by a few people 6-8 weeks ago. It was heavily in some of the infectious disease, a few infectious disease people and a few industrial hygiene people. And the data to suggest that those have some efficacy, similar to a bad mask, was for a product that went from your forehead to below your chin, back to your ears, almost basically touching your ears. That data is now being applied to little things that look like half of a duckbill that just barely cover the mouth and the underside of the nose, which is not supported. And people need to take responsibility for protecting themselves and others. I'll tell you one personal thing. When I look at somebody who's got either no mask or an ineffective mask on, I figure they're a double risk to me. Because one, they're not protecting me from them right now, but I also make the assumption that they're more likely to be carrying the virus because they're not protecting themselves from others either. So I have a real problem when I see that at stores or businesses or on crowded places.
Well, can I chime in?
Certainly.
Just to convey how important mask wearing is, I just want to relate some data. We did a study at Penn of over 2,000 healthcare workers who were, in the midst of the peak of the epidemic in Philadelphia, working with infected patients in the emergency room and the intensive care units. And we implemented a universal masking procedure, mandating masks for all our healthcare workers and visitors to the hospital, frequent hand washing and distancing where possible. And what we found is that only approximately 2% of our healthcare workers developed antibodies to the virus, indicating they'd been exposed to it. And that was during the same period that we were testing pregnant women coming to the hospital to deliver, not symptomatic from COVID, just coming to deliver their babies, presumably representative of the community around the hospital in the Philadelphia region. And at that time, 6% of those people had positive antibodies and were exposed to the virus. So the conclusion is that working in the hospital with sick people, as long as you're following the rules, is actually less dangerous than just living in the community outside the hospital, where likely people weren't masking and weren't following the rules as well. Every indication is that wearing masks really makes a difference. And I just want to underscore that point.
Thank you. Shifting back to discussion of vaccines, if I could, I appreciate the range of options that are being currently studied, Jonathan. But what's your view of the most promising vaccine development projects? And the inevitable question: could you give us any kind of time estimate as to when we may be able to benefit from them?
Yeah, well, let me say that first, I'm hopeful about the vaccines. I think that a number of preliminary data suggests that good chance they're going to work. That is, several of the vaccines have now been tried in non-human primates who have been then challenged with live virus, and they've been resistant to infection. That doesn't always translate to it working in people, but it's better than if it hadn't worked in non-human primates. It's encouraging. Also, the way the body's responding, as best we understand it so far, to infection suggests that immunity, is developed. Good antibodies, neutralizing antibodies that prevent the virus from growing in the laboratory, are produced by most people. And the immune system's memory, that is B cells that make those antibodies, seem to remember exposure, at least for a period of time, maybe not forever. With other coronaviruses, it's probably a year or two that you might remember the exposure. But also the T cells, a different arm of the immune system, seems to respond, remember. So my bet is that one of these vaccines is going to work. The many vaccines that are in late stage trials now are all related in that they're trying to produce an immune response to the proteins on the outside of the virus, mostly the so-called spike protein that binds to its receptor called ACE2 and is required for entry into the respiratory cells. Each tries to elicit that immune response with a slightly different trick. Either we give you the protein itself, so you make an antibody to it, or we give you a vector that encodes the protein, so you make the protein and then you make an antibody to it, or something like that. But those technical differences don't make a big difference in how likely it is to work. I will say that several of the leading candidates, DNA and RNA-based vaccines, have never before made a successful produced, a marketed vaccine. They have been used in the laboratory, they have been used in people, but never made it to market. So they're a little riskier. And that's the Moderna, the BioNTech, several of the other vaccine, Inovio. But actually, they elicit good immune responses. So on the one hand, it hasn't been done before, so you might not put it lead of the pack. On the other hand, the preclinical data is strong, so you might raise it up a little bit as a leader. I can't pick the winner. At this point, I think there's likely to be a half a dozen that will elicit good immune responses. That's my bet. And I think the differentiator is going to be side effects. If you're going to use it in 100 million or more people, it better have a really low side effect profile, because even a very rare side effect is too common, especially if you're giving it to healthy young people. So we're not going to know that till we do phase 3 trials with 30,000 people in each trial to pick up a low rate of a side effect. And that's why it is so critical that we do those trials before we give approval or push forward with backing one vaccine or another. All this world needs right now is to have people not believe the approval process any more than they do now. You all know about the anti-vaxxers, people who are reluctant to take vaccines anyway. We need to come forward with a vaccine that we have the very most confidence in. So I'm a big believer that we need to go through the process, do the phase 3 trials, don't rush it out for a election period. And I think that's going to take until early next year to know whether we have results on that. And then it's going to take some months to manufacture enough of it. So I wish I could say it was sooner, but I don't think it's going to be before a year that we have a lot of validated vaccine for the public in this country.
Thank you. So first of all, we did get a quick question here about MMR.
Yeah.
Is the MMR vaccine helpful at all?
Yeah, I saw that in the questionnaire. I meant to address it. I apologize. There have been a number of proposals that vaccines to various other things might elicit a sort of general immune response that would help you in fighting off this infection or other infections. And it's not crazy, that idea. There's some evidence out there that being primed with some other thing that stimulates your immune system makes you a little less sensitive to all sorts of different infections. But there's no good data that the MMR or other non-specific vaccines will actually reduce infection for this particular virus. So it's an interesting idea, but no good data yet to support it.
We just have a couple of minutes left before we get into the official Q&A section. So I have I know which is not a short question for you, Jonathan, but I just wanted to alert you to the time frame. And kind of the corollary to the question about vaccines is therapies. Do you have any insights for us as to what you see as promising treatments for COVID?
Yeah, I'll try to be quick. I appreciate the time check. I'll just say that one thing I'm excited about is that we've set up a screening procedure to screen all 35,000 approved FDA drugs that are in your pharmacy right now, and many of them generics that would be rapidly available if we knew they were. And we're lucky to have the ability to screen them in a laboratory where we can use live virus, a so-called biosafety level 3 laboratory, where everybody's suited up in an astronaut outfit so they don't infect themselves. And we have robots that can do the screening procedure. And that has identified several promising early agents. One of them, for instance, is cyclosporin that's widely used. And you'd think, it might knock down the immune system. That's how it's used in some ways. But it turns out the mechanism by which it seems to prevent the virus from replicating is not through its immune function, but through a different part of the molecule, something that it goes through a cyclophilin pathway. And so there are congeners of cyclosporin that retain this antiviral activity. And we have already approval for a clinical trial to test this in people. And there are at least 5 other candidates like that. The trick will actually be getting them through clinical trials with enough patients in a good study to know if we can believe the results or not. So there is some hope on the horizon with new therapeutics. And I know I'll be quick, but the other one you've heard a ton about these days is convalescent plasma. It's been all over the press recently. An old therapy of passive immunity. Take the antibodies from somebody who's recovering and give it to somebody who's sick. And thousands of patients in this country have gotten convalescent plasma. Let me just say the available data is mildly encouraging, but certainly doesn't prove that it's a panacea, despite what you hear on TV. And once again, I've been disappointed with the press coverage and with our own government officials in how they're massaging the data to sound more encouraging than it is. It's a therapy we're testing in a controlled, randomized trial. I think it's hopeful, but it's not going to be enough, and it's going to have a moderate effect at best.
Great, thank you. So JR, I was going to save the last word. Again, we got about 30 seconds. The other thing that's been in the press a lot in the last few days is the unfortunate wedding that occurred up in Millinocket. And my question really is are there any insights that we've seen from that outbreak that we want to make sure we bring home to our community?
I guess one insight is simply everybody in Maine, or at least many people around here, have been focusing so much on the risk from away. Forgetting that while the risk from people who reside here is statistically lower than someone who, say, just arrived from Georgia, that doesn't mean that we are not a risk to each other. And we need to protect each of us from each other. A second related piece of that is you have to choose your risks. What risks are acceptable and why. And I'll be very honest, I'm very sympathetic. I think a wedding is very different than a bunch of people getting together just to get together and drink. I mean, that's a sacred event. And so I feel terrible for the people involved. It does clearly show the issues related to, last time there was a question for Kate and I about super spreaders, and we each gave a different answer, and I forget which of us gave which. One of us talked about super spreader events, meaning a place where lots of people are in close proximity, laughing, speaking loudly, singing, often associated with alcohol to release inhibitions, but not necessarily. One of the biggest super spreader events was gym classes in Korea. The other definition of super spreaders are people who are not very sick or not sick at all, but have a very high viral load. And that's an individual. And sometimes they can be like that for days at a time. This was pretty clearly, when you look at the epidemiology of it, a combination of both. It was the type of event, but also if you looked at multiple people who were symptomatic, highly symptomatic within days of being infected, just 2 or 3 days, and very high percentage of those people have become symptomatic. The person who was spreading initially must have had very high viral loads without feeling sick, which is a lesson. You can't say, just because I feel fine, I'm not infectious. That could be any one of us.
Okay, well, I'm going to have to stop there. I think that's very helpful, and I really appreciate the information from this question. I think we got a good, very good, broad understanding of some subject matter that I certainly didn't have before. And I would like to shift gears to our moderator, Danielle. Do we have questions that have been forwarded to us?
Yes, we do. I believe we covered the MMR vaccine one, correct? Yes. All right, so another one came in. Of the vaccines being favorably tested, is there a sense of how long they will be effective?
I think the answer, unfortunately, is we don't know. We're just going to have to wait and see. It's similar to the thought about how long your own immune response, if you've recovered from the disease, might protect you. We just don't know yet. There was just a report of a patient who seems to have actually been infected twice when they sequenced the virus the first time and then the second time, and it was different, acquired at a different place. And it's the first good evidence that a person might be able to be infected twice, although they didn't get sick the second time, which means maybe their immune system was primed and protected them, but with an N of 1, it's hard to know. So the answer is we're going to have to wait and see.
And Chair here with another way of both answering, looking at that question, but also at vaccines in general that people have to be prepared for. There's 2 different types of protection we might get from a vaccine, and some vaccines give one, some give both. A vaccine can protect me from getting sick. It can protect me from the virus deeply invading my body. But I might still carry that vaccine on my that virus on my mucosal surfaces and be able to spread it. Or a vaccine might make me essentially unable to get enough of the virus in me and unable for the virus to replicate at all, in which case I can't spread the disease. Has some big public health implications. If the vaccine prevents you from spreading it, then the vaccine doesn't have to help everybody. For example, it doesn't have to be efficacious in elderly, which a lot of vaccines are not. But if the vaccine only protects the person who gets the vaccine, we'll need a higher efficacy for that, and it will take longer to bring the epidemic under control. And that's something we have no data about so far as I know.
All right, another question that came in. Maine has a 14-day quarantine period for people arriving from many states. Could it be shorter and still be protective? If not, should it be longer?
If it was made shorter, it would be, this is what I talked earlier about, you choose your risks and how much risk reduction you want. If we made it 10 days, we would probably catch about 90% of the cases. If we made it 7 days, we'd probably catch about 80%. Some other states allow you a week and a test at the end of the week, which will probably catch pretty close to 100%. They're all valid strategies. It depends on how high a level of risk reduction you want for what cost.
All right. Given the large numbers of critically ill COVID patients, do we have any better understanding of sepsis syndrome in otherwise young and healthy individuals?
Anyone want to take that?
I'm not sure exactly what you mean by sepsis syndrome. There is a syndrome that's different in kids than in adults of hyperactivation of the immune system that makes people extremely sick. In adults, we tend to refer to it as cytokine release syndrome, where you seem to have your immune system secreting a lot of inflammatory proteins or cytokines that make people extremely ill, even if it's not the virus itself at that point that's seeming to make those people sick. And many of the clinical trials ongoing right now are targeted at damping down that hyperimmune response. In kids, there's a different syndrome where their immune system seems to go awry. That's even less well understood. But they're both a little different than sepsis, which we normally think of in response to bacterial infections, also related to cytokines, but then the body's different organs start shutting down in response to those. So clinically, it looks a little different, but I suspect that's what you're referring to in the question.
All right, and the final question that we have is, how are volunteers being recruited for the clinical trials of vaccines or other treatments? Do they represent the general population?
Yeah, that's a terrific question. I see it's from Patricia Hand. Thanks for the question. Most of the large trials are trying to enroll a broad, diverse group of people. The initial safety studies were done in healthy, mostly young volunteers, but the phase 3 trials want to encompass elderly people, people of all races, and much broader and inclusive group. Otherwise, we won't know whether it's reasonable to use it in those groups. So that is the goal of the phase 3 trials. And if I can, I just will add, all of the research studies that I talked about today, all of the things we stood up very quickly, couldn't have happened without philanthropy. We didn't have government funding to do any of this work. All of our health systems, including I'm sure your hospital, have been stressed by this pandemic, and there wasn't a lot of spare money to go around. And I'm just incredibly grateful to those who stepped up and allowed us to get our work underway. I'll call out Martha and Wistar Morris, who may be on the call. They responded, amongst many, many others. And I'll just add how lucky we are to have such a great local hospital here on MDI, and hearing how all the information JR is able to contribute, and Art as well. Thank you guys very much.
Well, thank you. And, one of the things that we focused on was we focused a lot on research, and we focused a lot on the local MDI environment. Since we do have a couple of minutes, I know that hospital at the University of Pennsylvania is also a major medical center in the city. And what's the greatest well, I shouldn't say it that way. Where do you see your biggest challenges in meeting the needs locally?
Yeah, well, it's been a very stressful time, as you well know. And, we actually have 6 hospitals in the region, HUP, or the Hospital University of Pennsylvania, as the flagship hospital. Princeton Hospital in Princeton, New Jersey was actually the hardest hit early on because it's located fairly close to New York City, which had, so many cases. And the biggest issues we've had more recently have been making sure we're caring for everybody with other diseases. People still get cancer. People still have heart attacks. And with so much effort and attention appropriately diverted to COVID, it's been critical that we still maintain our other efforts. And the worry that people are staying home with other symptoms because they're afraid to come to the hospital, is something we've had to respond to. And the response has been really unprecedented, and I think will long outlive this pandemic. It's going to change the delivery of care forever. We moved to telemedicine in a way that we've only talked about for years, but in a month, suddenly we were seeing thousands and thousands of patients through Zoom and through other approaches, which is great for many patients. A lot easier than facing the traffic in downtown Philadelphia and trying to get into the hospital. But it raises its challenges and IT challenges and getting things to the patients. We had a whole crew of people delivering care at home, delivering chemotherapy at home, trying to get medicines to everybody. So the logistical challenges have been and continue to be just enormous. And then finally, burnout. People have been separated from their families, living in hotels. We rented out a whole hotel so that doctors could be, nurses could be at work, but not exposing their family members. They've been going, 24/7. And this is dragging out, and the whole country is burning out, I think. But the healthcare workers in particular have been under tremendous strain. And we've set up all sorts of apps that allow people to get mental health. As we can get care through Zoom, they can get some mental health and find resources for themselves. So trying to care for our healthcare workers has been an important challenge also.
Great. Well, thank you. And I know from the firsthand experience that I actually had the benefit of a visit there recently, and I've been amazed locally as we watched our healthcare delivery system transform overnight, and then multiply it. I want to think about how many X times to changing the way care is approached at a major academic center. It really is amazing, and we're also grateful for the team that's doing that here. But thank you. I want to thank you both so much for joining us this afternoon. And I also want to thank Penn Medicine for helping to make your time available to us today. It's great.
We love the connection to MDI Hospital, and our residents really value their experiences here. So thank you very much.
Yep. So I do want to again thank our community for the commitment that you've made, and just want to reassure you that MDI Hospital continues to be totally committed to the safety of our community, and in providing as much information and support and education as possible to mitigate the spread of this disease. And again, we can't do it. We cannot do it without you. We need your vigilance. We need your support of one another, and we need your awareness of the need for better testing and treatments and vaccines. And also, I should add, and I also want to thank our supporters in the community, because as was mentioned at Penn, the support that we've received has helped us significantly to do the things that we've been able to do. The incidents, we're very fortunate here that the incidence of COVID-19 in our community remains very low. But it only will be able to do that if we continue to work together. So again, thank you so much for joining us today and for your commitment to your health and the health of our community. and we will continue to keep you informed as the situation develops. And we appreciate your participation in this important discussion today. So again, thank you, and thank you again for your time. Have a good afternoon.